There is very little science in my talk. Most of what I'm going to talk about is really just a personal account of my time in Sierra Leone and really the challenges that we face in trying to integrate laboratory support and real-time sequencing into the epidemic. Now I realize this phrase is particularly offensive and you might even say somewhat dangerous to put up on a slide in front of a room full of people who are computational biologists. But I think in the case of the work that we did, I think it's probably better to say these were symbionts. Basically, we would not be able to do our work without the help of the computational biologists who helped us, and I am eternally grateful for all of you who were involved. So I'd just like to say a personal thanks to everyone who helped. I'd also just like to point out that actually Ebola virus and genomics and sequencing is a completely new thing to me. My natural habitat is in Addenbrooke's hospital. It is not in Africa. It's certainly not in doing field virology. And if you take all the papers that we've published in the last 15 years and you generate a word cloud, what you see is quite clearly we work on norovirus replication. My lab does pathogenesis, small animal models. We do proteomics and reverse genetics. We do not do sequencing. We never have done. And we certainly don't do field virology. But through a sort of random series of events, which I'll try to describe, I ended up spending approximately 6 months in Sierra Leone working as part of the Ebola response, and several other members of my lab were also involved in providing lab support. So we've had some very nice talks today about Ebola virus sequencing and the outbreak itself. My interest in the outbreak came about in July 2014. As a virologist, I had no interest in working on Ebola virus. I'll be honest. I think it's a difficult virus to work with. It's incredibly challenging to gain any biological insight given the conditions in which you have to work with these viruses. However, like probably many of you in the audience, I recognized very early on in the epidemic that this was not something that we had seen before. And this epidemic wasn't going to go away. So I made a decision in July 14 that this is something I'd like to offer my help. And I phoned a number of people and said, is my skill set useful? The answer was effectively no. I wasn't a clinician. Nobody really wanted any help. But I was convinced, and others have indicated, there was a real diagnostic bottleneck. So the time taken from a patient presenting with symptoms to being tested was very long, and I was confident that with our molecular skills that we could contribute, and that other molecular virologists from an academic background, such as myself, could also contribute. So eventually, a few months later, I managed to get in contact with European Mobile Labs and Public Health England, who were deploying teams. This delay was actually quite useful. It gave me some time to convince my wife and kids that this wasn't a completely stupid idea and it was going to be okay. We were deployed, sent to Porton Down for a 5-day training period in October 2014, and then we were being deployed to Sierra Leone in November 2014. I was the team lead of a group of scientists who were going into Makeni to establish a diagnostic facility, one of the first diagnostic facilities in the northern districts of Sierra Leone. This was the plan of the treatment center as it was shown to us. So here you have the red zone where the patients are housed, the low risk zone and the undetermined risk. This is effectively just admin and the canteen. If you squint quite carefully, you can see the laboratory, which seems, for reasons that I still don't quite understand, they decided to build the laboratory bridging the red zone and the green zone. So the windows would open up into the red zone. I don't really understand why that occurred. We never really opened the windows. And just to point out, this is the lab store. This was eventually where we ended up with our sequencing tent, so we were right next door to the lab. This was the picture. And at the time, you've seen this picture, Christian put this up. It's a particularly harrowing account of the situation on the ground in Makeni at that time of the epidemic. It was a particularly desperate situation. There was an urgent need for diagnostic support. So off we head, our team of 10 scientists, and we turn up to the treatment center. Effectively, this is what we turned up to. It was essentially a building site, a very active and very busy building site. And as you can clearly see from our team, this is me and our team of 10 biomedical and clinical scientists. There was one other academic in here. We are clearly lab scientists. We're not laborers. We're not built for operating on a building site. But the first few weeks were essentially set around establishing the laboratory. We made the decision that the lab was going to open before the treatment center was going to receive patients, and we would be able to offer diagnostic support to the surrounding districts. So sadly, we had 3 of these trucks to offload manually. We did okay. We found everything. We got everything together. We set up the workflow, and the laboratory opened in and around December the 13th. This was significantly later than we'd hoped, but it was the best we could do it in order to maintain the safety procedures and to enable the workflow was fully operational. Now at that time, once we'd set up the workflow and started to process samples, as you can imagine as an academic, just processing 100 diagnostic samples a day becomes a little bit monotonous. We were doing manual RNA extractions, so after your 3rd or 4th batch of RNA extractions, it starts to drive your mind a little bit mad. So I started to think about other things that we might be able to help with, or my collaborators or friends in the virology field might be able to help with to contribute to the outbreak. At the time, this coincided with Jeremy Farrar visiting Sierra Leone. I'm a Wellcome Trust-funded scientist, so I did what most people do. I sent him a message on Twitter. I said, you should come and visit the laboratory, and we arranged a meeting. And we had a discussion, myself, Tim Brooks from Public Health England, and Andy Simpson, about different possibilities. What are the real needs on the ground? What do we really need to be doing as scientists, and how can we contribute? And as we've heard already, ourselves and a number of other people really identified a clear need for in-country genetic sequencing capability. The primary aim being really to identify and confirm epidemiological links in a time frame that was going to be useful for individuals on the ground. We also wanted to overcome some of the bottlenecks of generating viral sequences. Christian and those guys at Broad had done a fantastic job at getting samples out, but that pathway was getting increasingly difficult. The government had shut down on moving samples. But it also became very clear to me that we were complete novices to this field. We were not going to be able to compete with the professional sequencers. So our biggest contribution could be simply just generating this data and giving it to people who could really make some sense of it. So the idea was to try and move a sequencer to Sierra Leone, generate data, and release it to the scientific community who could really use this information properly. In the back of my mind, however, I thought if we were going to invest all this time and effort, we should be thinking about longer-term capacity building in the area of genomics. And in fact, Nathan, who's here in the audience, and Pardis wrote a very nice commentary on this and really highlighted something very important, that during the early phase of the epidemic, we had a number of sequences that were released, but as the number of cases dramatically increased, the amount of sequence data that was available to the community was minimal. So there was a real bottleneck, and we decided that that it would be wise to try to move a sequencer to Sierra Leone and do this. Now, I was quite naive, I would say. Having never done this before, I figured it wouldn't be that hard. How hard can it be? It was difficult. I returned home on the 23rd of December, and when all the offices opened in early January, I think I was told on the 5th of January, I had until the 9th of January to submit a grant application to the Wellcome Trust for a sequencer and all the infrastructure that we would require. And the general plan was that we were going to take 2,000 Ebola virus positive samples, and we were going to take extracts all the way through to sequencing in country, in the Ebola treatment center. So therefore, we needed to produce a grant and cost up all the apparatus and all the infrastructure that we require. Again, just to reiterate, we were complete novices to this area. We'd never operated a sequencer. We don't have one in our lab. We just send things out. But we had to select a platform for this work. So the sorts of questions that we were asking ourselves, what sort of platform is going to be suitable for doing this work? Of course, you need something that's robust, very robust and tolerant of very extreme environments. The tent we would be operating in had an air conditioner. It kind of worked most of the time, but it still hit 30 to 35 degrees in that tent when when it was during the dry season. We needed something that was easy to install, so it was capable of working out of the box. No company would send an engineer to do the installation, so we had to do it ourselves. We needed something that was low maintenance, that wasn't going to freak out every time an insect crawled over the top of it, or some dust got into it, or somebody banged into it. But at the same time, we need to make sure that we were generating data that would be useful to the field. There's no point generating crap if nobody's going to believe it. So we were trying to balance all these various issues. We wanted to do reasonably high throughput. We didn't want to do 100 samples. As I said, we thought there was a real need to produce a reference data set for people in the field to use. And we needed a really simple and robust workflow. We had never done next generation sequencing. It was myself and my postdoc who was going to do this. We are skilled molecular biologists, but we needed to be able to be confident that this would work. And last but not least, it was something that was very crucial, is we needed a company who was really willing to engage with this project and make it work. So this is supplier one, who will remain nameless. List price of the machine they offered us, £67,000, 568. Okay. The University of Cambridge price, if we were to buy one and have it in Cambridge, was 62,834. You know, a small discount, which is okay. We made it very clear what we were doing, what this sequencer was going to be used for. However, as soon as we mentioned that we were taking this thing to Sierra Leone, all of a sudden this price jumps, okay, £80,000 to just simply for us to move that sequencer. We would take it, they didn't have to move it, we had to move that to Sierra Leone. We would get a 2-week training course at the pricely sum of £15,000, but we would not have an engineer help us install it. And it was made very clear to us that, in fact, if we installed this machine, there was no warranty. So as you can imagine, this was a once-in-a-lifetime offer that was, we could hardly refuse, but we did. And we ended up going with an ion torrent, an AMPLISEQ, ion chef, ion PGM workflow. And I would say that without doubt, this is probably one of the best decisions we made in hindsight. These things have been absolutely robust and rock solid. We've had no problems whatsoever, touch wood. They've been carried around, moved around in the back of pickups, carried over fences on people's heads, and they still work straight out of the box. I'll describe the workflow in a bit more detail in a second, but essentially we use an amplicon-based sequencing which relies on AMPLISEQ. This was a largely untested Ebola AMPLISEQ workflow, but it was relatively simple. It took 2.5 to 5 hours, depending on how many samples. It was a liquid handling robot which then takes the libraries and generates the templates and loads the chips, and then we would put them onto the ion PGM sequencer that would, each run would take around about 2.5 hours. So we reckoned we'd be able to prepare 25 samples per 318 chip, and we'd run 50 samples per day. That was the plan. So we submitted the grant 9th of January. I had 5 days to write this thing. And then in that period, it's sort of the reality of the situation. I spoke to some more what you would call professional sequencers, and the reality of what we'd agreed to do, it kind of started to hit home. But I thought that there was no way anybody was ever going to give us this money. You know, I had no background in this field at all. The grant was written in 4 days until the 9th, until the end of February, when an award letter turned up from the Wellcome Trust. And this is, to say, one of the rare occasions where you get an award letter with a sort of sense of dread almost. I had convinced myself that this wasn't going to be funded, and I could go about, you know, we'd done our piece. I could go about my life as normal. So there was a period of really thinking about whether this was something I really wanted to do. Okay, we are not a sequencing laboratory. We do pathogenesis. This was not an area I was familiar with. But did we really want to do this? Why isn't somebody else better qualified doing it? It turns out other people were and trying, okay, and other people did manage to do this, but I wasn't aware at the time. And there was a real concern what was going to happen to my lab when I was sat in a tent in Sierra Leone. I have 15 people in my lab. I'd like to think I play some role. Probably I don't. But in reality, what happened was my wife convinced me that this was important, that if I didn't take this money and I didn't do this work, I'd be upset with myself. And so I did what every good husband should do. I listened to my wife. we accepted the grant, and we had a training period in Glasgow. We had 5-day training on the AMPLISEQ Iron Chef workflow. So 3 days training on ion torrent sequencing and how to run AMPLISEQ and do the process, and 1.5 days training on the Iron Chef and the PGM. This includes how to install them, how to service them, and how to repair them. Now, when you open these things, I'm no engineer. It turns out the engineer who was training us was no real engineer. He was an environmental biologist. These things are incredibly simple. When you open them up, there are things that just bolt in and out. To change and maintain these things in the absence of an engineer is actually relatively straightforward. So at the end of these 5 days, or 4.5 days, we left with a bit more confidence that actually, if something broke, we'd be okay. I felt that we could do it. The AMPLISEQ sequencing workflow as well, as a molecular biologist, when I read the bump that the company had given me and thought, there's no way this is going to work. This is too good to be true. It seems too simple. There's some catch. Essentially, it's a multiplex PCR that relies on small amplicons. Because it's small amplicons, it's ideally suited to samples that have maybe been prepared in emergency response diagnostic labs that haven't been prepared, haven't been stored effectively, so been stored at 4 degrees C for maybe 2 to 3 weeks. So it's very tolerant of degraded samples. It doesn't require a lot of nucleic acid, and essentially we do a single RT and then 2 multiplex PCRs, which is simply then combined, and the assay just requires you to add everything to one tube. It's relatively simple. It's incredibly robust. We've been able to take people who have never done molecular biology and get them to do this and run the sequencer, do the whole workflow. Never done molecular biology and never held a pipette, and they can do this. We could get full genomes from samples with a CT of around about 35. It's rare that you would get an Ebola virus sample with a CT of 35, but we had a few. But essentially, the workflow looks something like this. The libraries would be placed onto an Iron Chef, and this takes about 10 hours to generate and load the chips before then subsequently being run on the sequencer. We estimate it would take 24 to 48 hours from a sample arriving in the laboratory to be able to generate a full genome. But sadly, we had no opportunity to validate this pipeline, and the company had no validatory data either. There was Pasteur Institute were using this panel, but they would not release the data to the company to check and update it. So we were doing this blind somewhat. We purchased all the equipment. This is kind of a shopping list of the things that we bought. Total cost in and around £300,000. The challenging items, I would say, are the fact that the PGM requires nitrogen gas canisters and molecular grade water. Nitrogen gas, it turns out that you cannot purchase a nitrogen gas canister in the UK. You have to rent one, and then you have to accidentally sneak it on a plane that's going to Sierra Leone and continuously pay rent. But we could buy some from Germany and fly them over and then fly them back out to Sierra Leone. The other thing, so these are personal distillers that we bought off Amazon, which are great things. Actually, you take these distillers and you put the distilled water onto these portable milliQ machines. It works really well. And the mini PCR machines were invaluable at the start of our sequencing. These were the only machines we had that would get down to sort of 18 degrees. The fancy real-time PCR machines we had would not. So we bought everything. We shipped it all to Cambridge. We unpacked it. We tested everything. We then repacked it. Myself, my postdoc, and my lab manager put it all onto pallets, and we wrapped it up. We've become quite good at pallet wrapping, so if anybody needs any help moving laboratories, we know how to do this. This is all the wet reagents in my beer fridge in the garage. The wet reagents we decided to take with us in luggage. First trip, £50,000 worth of wet reagents. We had 8 pallets. So then we put them on a truck and we sent them off. DFID kindly offered, so the Department for International Development, were offering to ship these things out for us, which was really invaluable. We got on a flight and flew out before the flight landed. And this, as you can see from Armando's very smiley face, is the truck arriving intact. All the equipment arrived. It all managed to get there on time. And this is the tent that we were decanting into. It was essentially a storage tent. that had air conditioning in, just a storage tent for the laboratory. With these sorts of things, this is just a video of us building benches and unpacking all this stuff, but in terms of getting the stuff off the truck into the treatment center, you may have seen that the treatment centers all have 6-foot high wire fences, and you walk through bleach to get through. So imagine carrying these liquid handling robots at head height through the bleach. One did fall, it was caught. But as you can see by the fact that we were smiling at the end of this, we were pleased that we actually got the machines, at least got them to the tent and got them unpacked. Whether we could get the workflow operational and maintain a sequencing service was another thing altogether. What we ended up with was a relatively nice lab, I have to say. It wasn't that bad working in there. We generated walls from boxes so we could segregate reagent prep area, PCR areas, and the sequencing area. We had 2 PCR cabinets, 2 of these things. This is the Iron Chef liquid handling robot, the PGM, and one of the 6 canisters of nitrogen gas that we had. And we set about trying to sequence samples. That's what we were there to do. So in terms of sample access, how do we get hold of these samples? As I said, I had been there twice already for probably in and around 2 months by this point, working with Public Health England, establishing their diagnostic laboratory and running diagnostics in their laboratory. Now I was here as University of Cambridge, outside of Public Health England. But it was a collaboration. The only reason I was there was because of this link with Public Health England, and they wanted to sequence. So our primary source of samples was via the collaboration with PHE, and we were to have access to samples from Kerrytown, Port Loko, and McKenny. But this came with a bunch of challenges, I should say. So we have to remember that these laboratories were established as emergency response diagnostic labs. They were not there for research support. So the SOPs were not designed to have samples nicely catalogued and nicely stored, separated positive and negative samples. Things were thrown in bags into a fridge and left. And in fact, the vast majority of cases at the start of the epidemic, all the extracts were thrown away. So thousands and thousands of nucleic acid extracts were thrown, and only the plasmas were kept. The extracts that were kept were not collated. And this is one huge bag of probably 2,000 samples that we rescued from one of the labs. Essentially just thousands and thousands of tubes thrown into bags, positives and negatives. We just spent weeks just going through this stuff, trying to find the positive samples. The biggest challenge, I guess, was the fact that one day in June, we turned up to the lab to find that everything had gone. Public Health England had done the right thing and had moved they had an opportunity to move their samples from Sierra Leone back to the UK, and they did that. What they did not do was tell us they were doing it. So my postdoc, I had gone back to the UK, he sends me a message and says there's no samples. We'd sequenced, we'd processed maybe 300-400 samples at the time, but we were only 1 month in at that point. So we ended up with a sequencer in Sierra Leone and no samples to work with and all this reagents and all this. And it was a bit of a stressful time. Thankfully, we managed to make friends with the European mobile labs and the Dutch mobile labs who were still operational in Sierra Leone at the time. They were very helpful, provided us with samples. This is us collecting samples from Hastings, and these are the Nigerian mobile laboratory that was based in Cambia, who we worked very closely with. We actually offered sequencing to anyone who was operational within Sierra Leone at that time. Any diagnostic laboratory who was operational, we offered to take their samples, sequence them for free, and give them the raw data, all the analyzed data back for them to do what they wanted. Okay? We were here to provide a service. That was the only thing we had in mind. Sadly, nobody took us up on that offer. And actually, there were several examples of studies that I knew were ongoing, where samples were in country that I knew we could sequence within a few days. We're talking 100 samples. We could do that in 4 days, no problem. But the scientists involved in those projects decided it was better for them to store them, ship them out, and sequence them. Some of these things are still, the data still hasn't made the light of day. I don't know where it is. And sadly, many samples were frozen. Were destroyed. Some laboratories had SOPs that said that the samples, the extracts, and the plasma should be destroyed after the samples were confirmed. But we had samples, we started processing data, generating sequencing. So when we come to data analysis, as I said, we are a pathogenesis lab. We do reverse genetics and proteomics. We've got no computational expertise. So who was going to do this data analysis? Our original plan was simply to upload this data back to the Sanger Center, and our research parasite friends who are based here were going to analyze this data and integrate the data with Andrew and Gittes at the University of Edinburgh. And this is how we started. Okay, we started trying to send data back. The reality of doing this was difficult. So the only internet access we had was a Wi-Fi dongle, which at best might be 3G, at best. So the upload speeds were very poor. The file sizes were big, 2 to 5 gigs per run, 2 runs per day sometimes. So the only solution was really to move these guys to the tent. So we have Mi-Fan and Matt Cotton, who came to the tent and worked in Sierra Leone. In fact, these guys ran the sequencing service for a period of 3 months. As you can see, Matt brought his flat cap with him all the way from the UK and kept it on inside the tent. But they ran it, and without them, we wouldn't have been able to do any of this work. They were essentially able to assemble the data as it came off the machine and making the files much smaller, which we were then able to send back to Gittes and Andrew for the subsequent analysis. So how did we integrate with the response? Well, this was not as straightforward as I thought it would be. A number of questions became obvious early on. So who needs to see this information? So the obvious things are WHO, who are not on this list, but WHO, CDC, the Ministry of Health, and any of the public health responders who were part of the epidemic. But what do we give them? You know, what format, what information do they really need to see? If we start waving phylogenetic trees in their faces, for many of these people, it's not going to mean anything. You need to give them something that is useful. The first few months, we were generating reports that were being sent to the chief medical officer and the district medical officer, and we would say make general comments about movement of viruses and transmission chains and lineages of viruses. But it wasn't clear to us if that information was going any further. And it turned out it wasn't. And it turned out that when we actually spoke to many people in the Ministry of Health, that it was seen that the sequencing data was purely of academic value. They really didn't need it. This information wasn't going anywhere. I guess a big breakthrough came when we met this lady, Damari Naidu, who was, I should say, I did try to convince these guys that sequencing was important, but my voice didn't carry much weight. This lady, Damari Naidu, who worked for WHO, she sat at the interface between us and the ministry and was able to coordinate everyone and make them realize the value of this data. And we formalized the integration a bit better in July 15, where we established what was officially termed the Ebola Outbreak Sequencing Support Service, which was supported by CDC, bought some reagents, and DFID helped support it as well. And basically, any new samples, new cases would be brought to us. We would sequence them within 24 to 48 hours. We'd assemble the data in country and we'd ship it back to Andrew, Gittes, Paul's team. We'd then produce a report in collaboration with the epidemiologists. We'd have a teleconference to discuss the interpretation of the data. Then a report would be produced and released. And I think in the end, this whole process worked quite effectively once after a few teething problems, and we were able to deal with all the flare-ups and all the recent cases that appeared in Sierra Leone at the tail end of the epidemic. So what did we produce? Basically, lots of Ebola virus genomes. I think probably over 600 in total. I think Matt told me there's a few more coming, possibly. And some of these genomes were done particularly quickly. Okay. I guess we were trying to provide contextual reference sequences to others in the field who needed access to this data. And I'll speak about data access in a second. We were trying to share the information with people in Guinea and Liberia as fast as we could. We did contribute to the TKM Ebola clinical trial, both in terms of laboratory support and sequencing support. Some of that's published. And there's some ongoing studies on particular clusters of infection where the clinical disease was let's just say this particular cluster, there was a high degree of hemorrhaging, sort of 70 to 80% hemorrhaging reported by the clinicians following a single introduction to one village where I think 80 to 90 people got infected. So there's some analysis to be done on this data. How did we do in terms of coverage? Well, I think we did okay. I get the impression from listening to everybody speak, particularly Gitz's comment, I think we did okay with sampling in Sierra Leone between the Broad and us and the other people who were operating the Chinese CDC. I think we did okay. What's still frustrating for me is the fact that, we were operational, we were in country in and by this point. We still have all these samples that disappeared, that weren't accessible to us. The ministry were telling the laboratories to give them to us. Some laboratories simply refused. Some laboratories continue to destroy the samples. We had, where possible, we were trying to do open data access. We were sharing this data, dumping data onto Virological, and with Richard and Trevor providing the data as well. So this real-time Ebola Nextflu tool was made available. And generating, Gittes and Andrew were generating these time-resolved phylogenetic trees. So we were producing reports to the Ministry of Health. But what did we really learn? Well, I'm probably the least qualified person in the room to talk about Ebola, what we really learned about Ebola virus sequencing, and we've had a few nice talks about this already. If you're really interested, you can read our paper in Virus Evolution. It contains all the sort of information. I would say, based on what the others have already talked about and had been published, probably we didn't see anything that surprising. Our biggest contribution was be able to do this in real time and try to integrate closely with the healthcare responders, and that was really what we were there to do. We saw evidence of cross-border movement despite border restrictions, as Gus has talked about. Geographical movement of viruses despite monitoring points being operational within the country. Sexual transmission and persistence of virus and evidence of slow evolutionary rates. We had a number of cases of this. And we have potential evidence of transmission via breast milk. Okay, this is only potential evidence. But one of the things, as I said, it became really important for us to do this in real time was during the tail end of the epidemic, and certainly with the flare-up cases, we were able to sequence these cases within 24 hours and produce a report as to the likely source of the infection. And during the end game of the epidemic, this is one particular example which was very important, which was the final case in Sierra Leone. On 15th of January, WHO declares the country free of Ebola. Then all of a sudden, a few hours later, a new case is identified. Sadly, all the international scientists from my lab were outside the country, but luckily we had trained 2 local scientists to do this. So this guy had never held a pipette, and this guy had never done any molecular biology, but in a period of months we were able to teach them to do this entire thing themselves. And I turned up just at the end to take a picture of them cleaning the machine as the sequencing was coming off. So they sequenced the virus within 24 hours, produced we then sent the data back. It was assembled and analysed by Gittes and Andrew. And quite clearly, this is evolutionary distance. This was the virus from January 2016. We could see quite clearly that the closest virus in all available sequences mapped to in and around November 2014. So this suggests that this flare-up came from re-emergence from a survivor. Sadly, we had no epi data to support this. Her partner at the time was not a known survivor, and he would not consent to testing. So we were unable to really confirm that this was a sexual transmission. So what did we learn as a result of all this effort and all this money? Well, in terms of open access data, funders do play an important role, and I'm glad now to see that they've taken a step forward and made a statement about this. This is the statement that's been released by a number of, including the Wellcome Trust, that in the context of a public health emergency of international concern, there is an imperative on all parties to make any information available that might have value in combating the crisis. Okay? This is a good step forward, but the problem is that it relies on buy-in from everybody involved. So it's not only the people receiving the money from the funders, but the laboratory partners that you interact with. In our case, there were lab partners who were not willing to enable us to do immediate data release, and they had very good reasons for that. The other thing, it became very clear to us, the practicalities of doing large-scale data release while you're in the middle of an outbreak such as this, it's difficult. There's a certain amount of effort required in data curation and getting hold of this metadata and producing the information in a usable format. And you have to balance the time that you spend actually doing sequencing and contributing to the outbreak with the need to really release the data, and if you're so interested, publish it. And in fact, we took this approach where we were trying to just basically contribute, and this is why it took us so long to publish. That was at the back of our mind. We really were too busy sequencing samples and trying to interact with the people on the ground to really worry too much about this. We did our best with data release. Sometimes it took longer than we'd hoped, but often this was technical issues rather than an active decision for us not to want to release the information. But being able to dump the data onto Virological was invaluable. This was one place that we felt we could put data without a huge amount of curation, and this is what we tried to do as much as possible. One thing that became very clear to me, and maybe naively I wasn't aware of this, is that people who work on academic research, so people like myself who are purely academics, we operate in very different arenas to those who work in the public health. And there are some people, many of whom are in the audience, we've had some very good examples over the last few days of where individuals bridge this sort of interaction where they work in public health, but they're also doing academic research. But we think about the currency that we are all measured by, so the metrics by which your success is determined. So in academia, you're essentially measured by publications and grants, okay? So whether you get tenure, whether you get promoted is essentially determined by whether you publish good papers and whether you bring in money. In public health, I apologize that there's probably a huge, big, long list here, but this is the only one I could think of. You're measured of service provision. This is your the main aim. During a public health emergency, this interface becomes very, very blurred. And in fact, academics who are operating in a public health emergency should bear this in mind and realize that their priorities need to change. And there were clear examples of people who we were interacting with, who were academic researchers, who had forgotten that they were part of an emergency response, and that the priority was to end this epidemic and provide this data rather than trying to publish a paper in a journal. And for me, I found that particularly frustrating. But the concerns that we need to deal with, and this is something that I know the community is thinking about, how do we enable people to share data, and how do we enable people to share samples while maintaining all their interests? Concerns are really around right to publish on the data, and the concerns around patient confidentiality and credit. The one thing that has become clear to me and has been really exemplified as part of the Ebola epidemic is that real-time sequencing on a short time scale is invaluable. And through data sharing, you can have a real impact on these sorts of outbreaks. But there are still significant rooms for improvement. I think Paul highlighted this quite nicely. So there are a number of bottlenecks. Sample prep is a problem. You still need, for highly virulent pathogens such as Ebola, you still need a certain amount of containment, a certain amount of infrastructure. You still need access to those materials. Data upload and analysis is terrible, and I know Nick is having issues with this at the minute with internet access, upload speeds. Mobile internet is a huge bottleneck. We also need field-ready equipment, reagents, and importantly, we need staff who are able to operate in these environments. We came at this from a complete standing start, which slowed us down. I think we did an okay job at getting things going, but if we were ready and trained and we had equipment that had been tested, we could have done this much faster. And one of the biggest bottlenecks for us was undoubtedly the need to have nitrogen gas canisters. I lost a lot of sleep over nitrogen gas canisters and the need for ultra-pure water. For the PGM machine, which is a fantastic machine, but in fact, it had its problems in this environment. It worked really well, but the need for gas, pure water and the initializations that take around 40 minutes or an hour and a half were problematic. The company have now released a new machine, which wasn't available to us, sadly, at the time, which is much more robust, doesn't require water, doesn't require nitrogen gas, and doesn't require an initialization. It's a 15-minute plug everything in, press go. And in fact, the Iron Chef, the liquid handling robot, will now do your AMPLISEQ libraries for you. It will then also generate the templates and load the chip. So by combining that AMPLISEQ and the Iron, the S5 and the S5 XL, I think in terms of hands-on time and skill requirements to do this sort of work, the bar has been brought down quite low. So just in the last few slides, just to talk about, what do you do with £500,000 worth of equipment, reagents and stuff that you've flown all the way to Sierra Leone at the end of the epidemic when they're closing the treatment center? We could just walk away and leave it. That probably would have been the easy thing to do. But what we decided to do was try to find a home for this. There is a university in McKinney. They don't have any biological sciences. They had a very nice house. This belongs to the president of Sierra Leone. We went in, they allowed us to use it. So we took all our equipment, we moved it all in to this building. We got all the equipment we could get hold of from the other laboratories that were closing down in Sierra Leone, brought it all together. We flew in some more stuff, benches, we had things made, and we've established an infectious disease research laboratory, trying to do some sort of infectious disease research. I don't really work on infection, I work on norovirus pathogenesis. This is a complete new area to me, but we are training local scientists and providing research support. We have other scientists who are coming to work in the facility, and we're enabling them to come and work there and undertake research programs within the facility. We're doing kind of low-hanging fruit stuff at the minute. So what can we do? We can get samples from the local hospitals. We have a biobank we've established, extracting and screening them for various viruses. We started to do some HIV sequencing, drug-resistant screens. This is just some data, a very new data, showing that there is some evidence of drug resistance in the local population. Hepatitis B virus sequencing. There's a reasonable amount of Hep B in the Sierra Leonean population, some worryingly high titers on some of these younger patients. We've started to set up a workflow for this. And more recently, we've tried to establish a ZIKA AMPLISEQ pipeline to have this in situ in West Africa if anyone wants to use it. This is raw data we just got a few weeks ago. It's essentially version 1 of an Ebola AMPLISEQ panel with cDNA at 3 different levels. I gather that clinical samples are in and around this area. This is no optimization, first go, same conditions that we do Ebola virus. And we can see that these are just contigs. You can see where things appear to be working reasonably well. There's some issue in and around CTs of 35 with the Brazilian isolates, but the AMPLISEQ, this is an iterative process. The company are now updating this panel, and it will be available, I gather, to people who want to use it. So just to finish, to really thank the people involved in the work. At any one time, we only really had 2 people, maybe 3 at the very most for short periods of time in Sierra Leone. These are the guys who actually ran the sequencer. I actually ran the sequencer myself. I wasn't just there taking pictures. I did actually do some work. I did, certainly for the first few months. And without these guys, you wouldn't be able to do this. Particularly want to thank Matt and Mifan for really stepping in when my postdoc resigned and got his own faculty position in DTU Vet halfway through the whole epidemic, which was great for him, but it put me in a rather difficult position. And just to finish by thanking the funders. All these guys contributed, and without which we would have been unable to do what we did. And with that, I'll finish and take any questions. Thank you.
One over here. So first of all, I love everything about this. I know how difficult it is to work. I can't imagine how you actually managed to do this, given the time frame is unbelievable. You mentioned many things to go forward and starting the lab and keeping capacity building and resources there, which I totally agree on too. The one thing that's missing though is money. So what is the plan for future funding, and would it maybe be you know, maybe should we actually think about doing a country-wide initiative, because we have a lab in Kenema, you are running with McKinney, maybe coming together would make a stronger partnership, but I just wanted to see what your take is on the funding
Absolutely.
situation.
You know, the sustainability of this is something that worries me. We were given 2 years' money from the Wellcome Trust, primarily just because I told them I couldn't, there's no way I was going to be able to employ anyone good for a period of 6 months. You know, we had to give somebody a contract for a certain period of time. So we have 2 years' money. We are trying to make the facility self-sustaining by bringing other researchers in and making them realize that, you know, this idea of parachute science, you fly in, you do your work and you fly out, or, you know, you just fly your samples out, to try and discourage that, and to bring the barrier to doing in-country research so low that it makes it easier. So, you know, we have now, as part of a collaboration with an Italian group, we have a 13-color flow cytometer, there's a liquid nitrogen generating plant, we have -80s, we have ELISA machines, so you can do a reasonable amount, but sustainability is the real problem. You know, I have money till next year. What do we do beyond this? And I think these longer term partnerships are really important. The problem is, I think many of the funders, I wouldn't say they don't like this stuff, but it's not, you know, supporting infrastructure and capacity building. It's something I think funders need to do more of. If an individual is doing research in a country which is underdeveloped, let's say, there should be a capacity building component of that research. It shouldn't simply be let's go in and take samples out. And I know Gus and yourselves and, you know, people have invested heavily in this, and it's not easy. The easy thing is to take the samples out and do the work in a nice shiny lab in the UK or the US. But I think the funders need to set a line in the ground and say, you know, this is no longer acceptable. If you're going to take samples from these countries, you need to show some capacity building component of that program of research.
So Ian, thanks, wonderful. I think a suggestion is that, because this has been in many evaluations, there is now a funders collaborative, which is called GLOBIT, that is looking at how to support funding for emerging disease outbreaks. And I think if there would be a joint something from everyone that has been involved to that group, that this infrastructure stuff really is needed, that maybe could be a forum, because it has the European Commission, Wellcome Trust, Gates, many of the big funders thinking about this.